Good morning, everybody. My name is Olga Kozhar. I'm a PhD student with Dr. Tobin Fever in Washington State University. And I'm going to talk about one of the projects that we've been working on for the last few years, about dynamics of Botrytis Cinerea colonization of raspberry flowers and fruit. Botrytis Cinerea is the fungus that causes one of the major diseases on small fruits, including raspberry, gray mold or fruit rot. So how does the botrytis infect red raspberry? Here is a raspberry plant, here is a raspberry flower, and we don't usually see symptoms on flowers. But when we see symptoms of botrytis infection is fruit. So all this grayish, fuzzy coverage on the surface of the fruit is a fungus that can easily spread from one fruit to another with wind and infect the whole field. So how it infects the plant? Based on the research that has been done around 30 years ago, the main hypothesis that we have right now is fungus infect flowers through the female part that's called carpel and stays inside flowers without any symptoms until fruit starts developing and ripening from these flowers. And this main hypothesis is used for the development of management strategies of the disease in fields until now. And the main management strategies are fungicide applications. So for example, here is a quote from Pacific Northwest Plant Disease Management Handbook that suggests growers to start spraying when flowers just get open, around 5% bloom, and continue applications every week or once in 10 days, depending on weather, and spray throughout the growing season. So such kind of management strategies are pretty extensive and can include from 5 till 10 fungicide applications in a 2 months growing period of raspberries. If you look at the development of red raspberries that you can see here on an x-axis, for example, starting from close green buds all the way to ripe fruit. So the calendar basis sprays that I just talked about briefly would look something like this. And also it's called full season fungicide programs. When fungicide applications, which is indicated in arrows here, applied from the opening flowers on a calendar basis through the whole season. However, there is not much research done on the effectiveness of different spray programs on raspberry fields, unless probably just one that was done in Ohio State University by Michael Ellis in 2008, when they actually tested different fungicide programs on raspberry fields. So the first program that they tested was a full season program, as described here. The second program was restricted only to bloom, so only flowers were treated with fungicides. Again, going back to the main hypothesis that flowers are the main source of infection later in the season. And the third program that was tested was pre-harvest program, when fungicide applications were concentrated only on fruit without flowers. In their results that they found, they saw that the disease incidence, so percentage of fruit that had gray mold on them from each of these fields, the highest level of disease incidence was with programs that were restricted only to flowers, only to bloom period, and was from 18 till 30% over 2 years of study. The 2 other programs, such as full season or pre-harvest, yielded approximately the same results. Sure. Full season and pre-harvest programs had similar results and yielded in much lower disease incidence from 1 till 10%. So this data tells us that the infection process of raspberries most likely is more complicated than just infection of flowers in the beginning of the season. And that's why we decided to design our project of botrytis colonization of red raspberry in field conditions here in northwestern Washington. So the main questions of this project were: how is infection related to flower and fruit development? Through which flower and fruit organs infection occurs? And how fungicide applications affect colonization process of red raspberry? So first, we looked at the development of red raspberries and divided the whole process in 7 developmental stages, starting from close green buds all the way to ripe fruit, in order to find out at which of these stages most of the colonization happening by the fungus. In order to find out through which organ fungus most frequently enters the plant, we also dissected all these developmental stages into their morphological parts, such as you can see here: carpel, stamen, petal, pedicule, receptacle, etc. This project was conducted over 2 seasons in 2015 and 2016 on unsprayed field at the WSU Research and Extension Center. A sampling was performed once a week, and all developmental stages that were present at every sampling date were collected and brought to the lab. So let's first look at the results of colonization of these different developmental stages. Here I'm going to show you results only from 2016, but the dynamics of the colonization between stages was the same between 2015 and 2016. So here on the x-axis, we have our 7 developmental stages, and on the y-axis, percent of samples that were colonized by the fungus for each stage. So first, if you look at the colonization of flowers, the first 3 stages, we see that the percentage of colonization of flowers was actually pretty low. No more than 15% of total sampled flowers over the whole season were colonized by the fungus. When fruit started developing from these flowers, the percentage of colonization significantly increased and reached about 40% at green fruit. And the letters above the bars just represent statistical significance differences between the colonization levels between stages. The level of colonization of unripe and ripe fruit again significantly increased and peaked at the ripe fruit stage, reaching almost 70% of total sampled fruit was colonized by the fungus. If you come back to our main hypothesis of flowers being the main source of infection of raspberry plants, and look at this graph, we would probably expect to see somehow a straight line after the level of colonization of flowers. So if assumes that flowers are the main source of infection, then the level of colonization of flowers and fruit would be probably similar or the same. Here we see a different picture that again shows us that infection process of raspberry plants by botrytis is most likely more complicated than that, and it can include multiple infection events and not just at flower stage. If you look at the results of colonization of different organs or morphological parts of flowers and fruit, again, on the x-axis, you see our developmental stages, starting from open flowers to ripe fruit, and on the y-axis, percent of samples that was colonized by the fungus at each morphological part for each of these stages. First of all, what we can see here, if you look at the red bars, that was the most frequently colonized organ, carpel, among all stages, from flowers all the way to ripe fruit. But what is interesting here as well, that when fruit started developing and maturing, we also can see that other morphological parts started significantly colonized by the fungus at later stages, such as stamen, sepal, and others. That again shows us that these other parts that get colonized later can be a potential source of direct infections. But that's something that we still exploring more. What is the role of these other morphological parts? So in the result of this project that we did over 2 seasons, the main conclusions were, first of all, limited colonization of flowers. The colonization by botrytis increased when fruit started developing and maturing. And also, carpels were the most frequently colonized parts, but when fruit starts developing and mature, other parts get colonized by the fungus. And the question is, so what is the role of these other parts in the infection of fruit? Since we noticed such a low level of colonization of flowers, especially in 2015, in 2016, we decided to do a pilot experiment, and one of the conventional plots that was sprayed with fungicides and just did a limited sampling of the same developmental stages and compare it to the level of colonization with our unsprayed plots. And we didn't find any significant differences in the colonization of flowers and fruit from sprayed plot. So in 2017, we decided to do a first year of the whole study of the effect of fungicide applications that we just did this past summer. For this project, we picked 2 fields that were about 70 miles apart that are conventionally treated with fungicides. Part of the field was sprayed with fungicides with a regular program, and part of each field was left without any fungicide application the whole season. Both of them were the same cultivar. The only difference between fields were their age. So field number 1 was planted in 2015. So the field was much younger and had a much dense canopy compared to field number 2 that was about 5 years old. In this project, we focused only on 3 stages: on flowers, green fruit, and ripe fruit that we collected for the analysis from all this, from 2 fields from both plots. If you look at the results, so on the x-axis, you can see the timeline of raspberry season in 2017. And also our developmental stages that we sampled at particular date. And on the y-axis, it's percent of samples that were colonized by the fungus. So first, let's look at the level of colonization from non-sprayed fields. Again, here we see that the level of colonization of these fertilized flowers was about 40–50%. And again, that's similar results that we got over the last 2 years of our studies of the same stage. The percentage of colonization of green fruit significantly increased and reached 100% from non-sprayed field. And the level of colonization of ripe fruit increased also and was also 100% from non-sprayed field. If you look at the results from sprayed fields, so by the time we sampled flowers from sprayed field, they already received 2 fungicide applications that you can see here with arrows and the names of the fungicides above the arrows. But the level percentage of colonization of these flowers was not different from non-sprayed field and was about also 50%. If you look at the colonization of green fruit from sprayed field, by the time we sampled, the field already received in total 4 fungicide applications. The level of colonization was significantly different from non-sprayed field, was about 80%. But again, 80% of green fruit was colonized by the fungus from that fields. And if you look at the colonization of ripe fruit from sprayed fields, by the time we sampled, it received total 5 fungicide applications through the whole season. And again, the percentage of colonization was about 90% of total sampled fruit had fungus inside. We also looked more closely to flowers and ripe fruit colonization by themselves. So here is the colonization of flower organs by Botrytis. Again, our organs on the x-axis, and the percentage samples that were colonized by the fungus were on the y-axis, spray versus non-spray. So first, if you look at the all morphological parts separately, we didn't see any significant differences in the level of colonization by the fungus between between sprayed versus non-sprayed fields. That again makes us question the effectiveness of protection of flowers. When we looked at the colonization of ripe fruit by Botrytis, I think this computer does not like my PowerPoint. Sorry about that. Okay, let's look at the colonization of ripe fruit by Botrytis. Now we're going to look at 2 fields separately, because the results were quite different between 2 fields. First, field 1, we have the morphological parts of our fruit on the x-axis and the percent of samples that was colonized for each of these morphological parts on the y-axis. So field number 1 didn't differ between sprayed versus non-sprayed in these parts, and the level of colonization of which is called druplet here, it's actually our fruit by itself. that we harvest was 100% from non-sprayed field and about 90% from sprayed field. If you look at the field number 2, we see the different picture. We see that level of colonization of especially drupelets, which is of the main interest of fruits that we harvest, was significantly lower from sprayed field comparing to non-sprayed field. One of the possible explanations why we have such a big differences between two fields, again, it can be their age. As I said in the beginning, field number 1 was a much younger field with much dense canopy. So our hypothesis is that because the plants had so many leaves on them, the effectiveness of coverage by fungicide sprays may be not as efficient of fruit as, for example, on field number 2 that had much less dense canopy. We also noticed on ripe fruit that, so here so far I was talking about colonization incidents. So we just counted how many fruit from total sample had fungus on them. But we also noticed that the amount of fungus from sprayed versus non-sprayed fruit significantly differed between between sprayed and non-sprayed field. So if you look at an average number of colonies per one ripe fruit, so from how many places fungus grew out of the fruit, again, two fields, field number 1 and field number 2, we see that the number of colonies per one ripe fruit was significantly lower from sprayed field, which is red bars, comparing to non-sprayed fields in both fields. And in field number 2, we see that the effect was even much stronger. So it was about 1 colony per ripe fruit from sprayed field, comparing to average of 6 colonies in ripe fruit from non-sprayed field, showing that fungicides do decrease colonization of ripe fruit. So in our conclusions, in addition to the previous findings from 2015 and 2016, that colonization of flowers was limited, it increased when the fruit developed and matured, that carpels most frequently colonize parts, but other parts colonize later that may facilitate in additional infection events later. And also this past summer, we found that fungicide applications don't seem to decrease botrytis colonization of flowers, but they do decrease colonizations on ripe fruit. So if we come back to our main hypothesis and our current management strategies with calendar sprays and full season programs, based on this data of last 3 years, it seems like the early sprays that we apply at flowers may not be as effective in protection and decreasing gray mold on a field. So if we could decrease the amount of sprays and maybe eliminate some of the early sprays, first of all, it would be much more economically efficient, it would save money, but also it would decrease fungicide resistance, which is a huge problem in the area because we have such extensive fungicide application problems. But in order to do that, we would need to do more studies. And I'm planning to repeat the experiment that I did last year, next year, and maybe include more fields to see if we get consistent results over multiple years and find when the fungicide applications can be eliminated. With that, I would like to thank you, WSU Small Fruit Pathology team, Washington Red Raspberry growers that collaborated in research, and of course our funding sources. Thank you, and I'll be happy to answer your questions.
All right. Excuse me. Anyway, all your formulas and everything didn't put into the picture of the so-called moisture effect, you know, of more causing of the fungus so you almost need to have what the weather is having to do with what you're speaking of. You follow me?
Mm-hmm.
All right.
So what's the effect of, the question is what's the effect of weather on colonization? We monitored weather over 2015 and 16, and as you may remember, 2015 was much drier and hotter. than 2016. And I can say that both temperature and moisture do affect the infection of botrytis.
Olga, I wanted to thank you for this report, which was great. I think that answers a lot of questions. But my question is really similar to Daryl's. I was wondering if you're thinking that those first two sprays are less critical than we've been thinking, would a grower want to make those sprays anyways if the weather is conducive to botrytis, or if you anticipate the weather is going to be conducive to botrytis?
Thank you, Tom. That's a good question. I would say, first of all, that we didn't have a conducive year of the disease yet for this last few years. So I would say, I don't know. But also, if you come back to the development, how plant develops and how it flowers, from what I was noticing over this last few years when I was watching flowers and fruit development, it seems that the window at which flower is open only 1 or 2 days. And we do apply fungicides once a week. So majority of flowers that get open and then get fertilized and become fruit remain non-sprayed, remain non-protected. So again, even if you have a conducive year, I don't think you would be able to spray every day. So again, still, I'm not sure how effective the fungicide applications would be on flowers, even if you had more conducive year.
I have a question. I saw you like have a chemical spray. You put the captain first and the switch later. Do you think you put the switch first and captain later will help? Yeah, captain is more like a contact fungicide, and the switch is systematic. Systematic means the plant will absorb and then have the resistance later.
So the question is, if we switched first to sprays. Mm-hmm. Mm-hmm. I'm not sure if that would affect, again, for the same reason I just said about development of the plant, and also we didn't see any differences in the colonization between sprayed versus non-sprayed, and also just that flowers open for such a short period of time. But we did not test that. Yes.
Do you theorize the same might apply to strawberries?
That
it might, would you theorize that the same factors might apply to strawberries?
I'm not sure. Based on the research that's been done on strawberries, in strawberries, it seems like flowers are important. Based on the research that I was reading before, in strawberry system, it seems like flowers seem to be important in the development of fruit rot. So I cannot say that that can be transferred to strawberries.
Just a clarification here. So in the relationship between strawberries and raspberries, what would you say is the physiological difference that differentiates them from responding to those early flower sprays?
Can you please repeat it?
Okay. So in relationship to strawberries and raspberries, what's the physiological flower difference that you hypothesize in this experiment is leading to the difference where we see very good efficacy on flower sprays in strawberries traditionally, and why is that not carrying over into raspberries based on?
Okay, so the question is, what is the physiological differences between strawberry and raspberry? I didn't study precisely strawberries by myself, so I guess I don't know what are the physiological differences. It can be, I don't know what the longevity of flowering as well of strawberries precisely. So that might be longer too. So I don't know.
I wanted to ask about biofungicides on those early applications. Have you looked at that? Because that would help you with your resistance management, give you an opportunity to populate that flower with a antagonist that would antagonize the pathogen. So have you looked at that as an early application, biofungicides?
Early applications of biofungicides?
Yes.
No, I have not looked at that. But that would be interesting to explore as well.
Yeah, 'cause that's what they do in the apple industry. They try to get it on the stigma before, like for fire blight, you get in and populate it before the pathogen gets there. So that would be a good opportunity at that point in time.
Yeah, I remember there have been studies done with trichoderma fungi, for example, and there are some papers that were saying that early applications of trichoderma fungus seem to decrease colonization, but I didn't see extensive research on that. So that's, I think it's still question how effective they are.
Yeah, it's not going to be up to the standard of the conventional, but it may give you some extra protection instead of waiting until you really have a problem. So.
Right. Thank you. Any questions? More?
Hi. Do you think that sprayer droplet size can make a difference, like an air blast versus a rear size, as far as getting in the flower and killing the botrytis and the effectiveness?
I think it might. And also, like, how well it covers flower and plant surface. So answer is yes.
The difference between like field 1 and field 2, were they different?
I believe that the same sprayer, like the same program, the same sprayer was used on 2 fields. So I don't think there was a difference in a droplet size because it was the same farm, the same equipment was used on both fields. Thank you very much.
Thank you.
Should I just pause it? Thank you.